would be Mike Mehan. There you are, Mike. All right, so Mike, thanks for volunteering to give a talk. Mike will talk to us about leveraging nested data frames in regulatory submissions. Mike, take it away.
Thank you. Can you guys see here and see my presentation okay?
Yes, we can.
Great. Hello, everyone. My name is Mike Mehan, and I am a staff bioinformatics scientist at Illumuna. And today I'll be talking about how you can use nested data frames to improve the traceability and reproducibility of analytical studies. So Illumuna is most well known for producing sequencing instruments, but there are some clinical markets in which we develop our own end-to-end solutions. And I've shown some of those here. These are typically screening or diagnostic assays. As a member of the bioinformatics team, we help in assay development, and then eventually, as we get them ready for regulatory submissions, we do a lot of the quantitation of analytical performance. A lot of these analyses require repeating very similar analytical performance analyses on multiple events, right? So you might have an assay that can detect multiple disease states, which I'll refer to as events. And then for each of those, you might have multiple instances of affected individuals, which I'll call positive samples. And then you may need to analyze those using different reagent lots to study lot-to-lot variability. So what this results in is having to perform a similar analysis pipeline under multiple assay conditions. And for today's talk, I'm going to show you some use cases of how nested data frames can help facilitate this. And to do that, I'm going to use a simulated limited detection data set. So the design that you'll see me refer to throughout the talk will have 3 different disease states, which I'll just arbitrarily call events, events 1, 2, and 3. And then for each of those, we'll have 3 affected individuals, which I'll call positive samples. And then we'll have 2 lots for each of those for 18 total experimental conditions. Limited detection, for those of you not familiar, this is something that you look at when you have a diagnostic assay. Ultimately, what you want to report on is what the analyte concentration is that gives you a detection rate where you can detect the disease state 95% of the time. And that's done by fitting a probit regression model to a concentration titration, and then inferring from this model what the concentration is that gives you a detection rate of 95%. So I'd like to start with a bit of a counterexample. So this is a coding pattern that I've used a lot in the past anytime I'm doing analysis under a lot of different conditions, right? So you might use lapplies or maps to iterate over all the different conditions in which you're doing your analysis, and then you do your analysis pipeline, right? So for this example that we've been discussing, we are filtering the experimental data based on the disease state, positive sample, and lot of interest. And then we're fitting that probit regression model, and then we're summarizing the results of the model, which in this case are our detection rates. And then you might package that up in a list and return this nested list structure. And this works fine, but it does have some limitations. One is it can be a little hard to interpret, right? Unless you document the structure well, it can be hard to understand what level is which if they're not properly named, and so collaboration can be a challenge. The other thing is getting data out of this can also be a bit clumsy, right? You have to recurse through this nested structure, get what you want, and then do this packaging on the way out so that you have either a data frame or some useful structure that you can use going forward. So what I'd like to show going forward is how nested data frames can address some of these limitations. So we all know what a data frame is, right? It looks and feels like a matrix, but it's actually a list, right? It's a list of vectors, and the vectors contain scalar values, strings or numerics or logical values. So a nested data frame it can be similar. It just extends upon this concept where instead of just having the columns be vectors, you can now have columns that are also lists. And in those lists, you can have more complex types like a GLM fit, or each entry can itself be another data frame. So if we go back to our example from before, right, if we were going to put that nested list example that I gave into a nested data frame, we'd start by creating a data frame that has all of our conditions, right? All combinations of 3 events, 3 positive samples, and 2 lots. So that gives us 18 rows, where each row corresponds to a particular study condition. And then for each row, we're just executing our analysis pipeline. We're starting with, we're filtering the experimental data to create a data frame, a training data set that corresponds to those conditions. And then we're fitting a probit model for those, and then putting the results of that probit model in a column in our data frame. And then for that model, we're extracting the detection rates. The difference is each of those results are now in their own column in this data frame, and we do that by using map, by mapping each previous step and creating a list that is stored as a column in the data frame for the next one. So this is what that looks like in practice, right? So you have a single data frame that has all of your study designs and your downstream analysis steps. So if we compare that to the previous example, the nested list, where it was a little hard to see what nested level corresponded to what, now we have named columns. So we can quickly see what column corresponds to which piece of analysis, and each row corresponds to a particular condition. So for the rest of the talk, I'm going to very rapidly go through a few use cases. These are going to come pretty fast, but I will at the end put a link in chat for a notebook that you guys can check out if you want to dig into them in more detail. So the first one is a pretty trivial example. Let's say that we want to quickly summarize the goodness of fit for our models. With a quick map, we can add the AIC to our data frame. And because the data frame already has our study design in it, we're just a few lines of ggplot away from quickly creating a plot that summarizes all the models in our study. The second example is a little more involved. So here, we might want to, in this example, we're trying to calculate the maximum LOD for each positive sample across lots. This is something that's typically done to give a conservative estimate of LOD. So the way that works is you first, from our large data frame, select the columns you care about, which are the study design and then the detection rates, which are the model summaries. And you can see that each of those is a 6x2 data frame. Then we unnest. And so what that does is it adds those 2 columns to our data frame, the detection rates and the concentrations that correspond to those. And then you get 6 times more rows because you've now expanded all those out to create a larger data frame. And from there, now we're just doing standard tidyverse group by and summarize, right? You get the LODs, which are the 95% detections. And now you have a table of showing what is the maximum LOD for each of these combinations. And from there, we can spread to create a sort of report-ready table of all of our conservative LODs. The last example that I want to show is with pmap. So pmap can be really powerful in this context. I use it quite a bit. The way I typically use it is I first define, if I have a piece of analysis that requires a lot of inputs, right? So requires, in this case, 5, the training data, the probit fit, and the study design. I'll create a function, and I'll define the arguments of that function to match the columns of the data frame. I do that for 2 reasons. One is it provides like some clear documentation, right? This function does this, and it uses these inputs from our data frame. The second is that pmap is from per. And it's a map function that is implemented to take a named list as the first argument. And because our nested data frames are named lists, we can just simply pass the entire data frame to pmap. It'll extract the columns from the data frame that we need, apply this function to each, and add a new column where each row corresponds to the output from those corresponding inputs. In this case, a plot that shows the probit fits, the training data, and then has the study info in the title. So I know that all came pretty fast, so apologies if that was a little hard to follow, but what I'd like to do here is just try to tie it all together for you. So what I'm showing here is the single data frame that I used to both simulate all the data for this analysis and do all the analysis, all the downstream analysis. So here I quickly simulate some regression coefficients for each event, and then I fold in the different positive samples and lots and jitter those to create some variability, insert a random seed for each training data set, and then using pmap, as I just showed you before, I can create a simulated training data set for each of these conditions. And then from there, we're doing our analysis. We're fitting a probit regression model, and we're doing detection rates. So what's important about this is I've now decoupled the reproducible nature of my analysis from a notebook, right? I no longer have to render a notebook to recreate all my data. I have this modular data frame that I can give to another analyst and say, okay, here's my inputs, here's my random seeds, here's a validated R package. Can you recreate and confirm that you get the same results? So I see that I'm up against my time, so I'll just quickly say here, the real benefits are, one, you're in a data frame, so you can use tidyverse to be very efficient. And the second is this nested data frame serves as like a little mini environment. So it forces you to stay organized and put every piece of, every input that you need to conduct your analysis in one place. And then it's now this one modular component that you can save for later or use for downstream analysis. So thank you for your attention, and I'll close there. And I'll look for a link in the chat. I'll drop you that notebook if you're interested in more. You can always contact me on Slack if you'd like to chat. Thank you.
Thanks, Mike, for that wonderful use case. I think there's some very nice comments here about how that changed people's lives, some of the use. So thank you again